Welcome back, class. So our last lecture video, we talked about a lot of ways that can be used to target specifically bacterial cells. But sometimes the infection you're trying to get isn't bacterial. You need to be able to treat other types of infections too. One of those types of infections might be a fungal infection. The thing with fungal cells is they are eukaryotic, just like our body is eukaryotic. So it's going to take a little bit of more careful focus to target an antifungal. One thing that fungal cells do have, though, that we don't have, is a cell wall. So if we can target their cell wall, that's a really good way to target a fungal infection without affecting our own cells. Mainly these drugs that we're looking at in this chapter discussion, at least, are targeting either the sterols in the plasma membrane of the fungal growth or the cell wall. So polyenes are going to target the sterols in a fungal plasma membrane. Azoles and allylamines are going to target the sterols in the plasma membranes. And echinocandins are going to interfere with the cell wall of that fungal cell. So azoles and allylamines are mainly going to be used as lotions to treat cutaneous infections, something like a ringworm or a jock itch. Sometimes they're used for systemic mycoses or something that's gotten into a little bit deeper into the body. And then echinocandins are going to lead to lysis of a cell wall or lysis of the cell because they don't have a functional cell wall. These are also used for skin infections like ringworm. A virus is a whole new mess to try to target because a virus is growing and replicating inside of the host cell. So you can't target a viral ribosome because it's using your ribosomes. And you can't target a virus's enzyme because it's using your enzymes. So it's really hard to target a virus without hurting the host. One thing we can do is accelerate their high mutation rate. In our own bodies, our bodies naturally make things called interferons. We'll come back and talk about them when we talk about chapter 16, our immune response. But interferons are proteins that our cells make when they're infected by a virus. And those interferons spread as kind of like a text message to the cells all around it to tell the cells around it, hey, there's a virus in the area, lock your doors, protect your kids, like shut down, batten down the hatches. So it stops the virus from spreading to new cells. One drug that is important to talk about are antiretroviral drugs. These are drugs that are used to treat HIV specifically. The thing about HIV is it's a little bit weird. So in our cells, we have DNA that is used to make RNA, and RNA is used to make proteins. In HIV, they're an RNA virus. They start with RNA. They don't start with DNA. They start with RNA. And then they use that RNA to make DNA. And then DNA makes RNA, and RNA makes protein. So they start with that extra little step, right? RNA to DNA. That's called reverse transcription, which is something that really only these retroviruses do. So that step of RNA being used to make DNA is a good step to target HIV because it's something that our cells don't need to be able to do. So we make these antiretrovirals that stop that synthesis of DNA from RNA, so the virus is not able to replicate. Some other targets of antiviral drugs would be receptors on host cells. So this is mainly important before the virus has gotten into your cell. If you can block those host cell receptors, then the virus isn't able to bind. I've seen a lot of HIV drugs that are used as block that receptor. If you are at risk of a particular viral infection, sometimes that is a treatment that you can use before being exposed. Some antiviral drugs are used to stop the viral DNA from integrating into the host DNA. So it doesn't necessarily stop the virus from existing, but it stops it from messing with your DNA. One is by inhibiting synthesis of viral RNA or DNA, and one is by blocking neuraminidase. Neuraminidase is something that a virus needs to use to get out of your cell so it can spread. So if you block neuraminidase, then the virus is trapped inside of that cell, so the infection is then contained. Next, we have protozoans. Protozoans can cause some infections. They tend to be slightly anaerobic, so we can block anaerobic metabolism and block them. Quinine is a drug that is used to block Plasmodium, a protozoan that causes a disease called malaria, although there has now developed a lot of resistance against quinine. So chloroquine has largely replaced quinine. Quinacrine is used to protect against the disease giardiasis, which is one that you can actually find in our area, especially in the summertime when the waters warm up. Giardiasis is a type of diarrhea that is caused by the protozoan Giardia lamblia. And then antihelminthic drugs are drugs that are used to kill helminths or worms, parasitic worms. We have niclosamide, which will kill tapeworms by inhibiting their ATP synthesis. Praziquantel, which kills tapeworms by altering their plasma membrane so it becomes more permeable and their cellular contents will leak. And we have mebendazole, which will inhibit the formation of microtubules, which means that those cells without the microtubules, they can't do endocytosis, so they can't bring in nutrients. So which component in the cell membrane of fungi is the most common target for antifungal action? Notice this is asking cell membrane, not cell wall. So what is the target in the cell membranes of fungal cells? One of the most widely used antivirals, acyclovir, inhibits DNA synthesis. Humans are also synthesizing DNA, so why would this drug be useful in treating viral infections? I want you to think of how rapidly a virus might be needing to synthesize itself, to copy itself, and how often a human cell needs to copy itself. So use that to kind of think about why that answer would be what it is. And then we'll come back and talk about testing drugs and drug resistance in our last part of this chapter. I'll see you all later. Bye.